So now we want to start to look at the two different major types of cells. The first type of cell is a prokaryotic cell, and this is by far much simpler than the eukaryotic cell. So we're going to spend a little bit of time on the prokaryotic cell and a lot of time on the eukaryotic cell. Again, what is our size range? Looking for most bacteria, most prokaryotic cells to be in the range of 0.1 to 10 microns in diameter, and that's quite a range. Most are going to be somewhere between 2 and 3, maybe 5 microns in diameter. Now remember that prokaryotic cell, that size is much smaller than the eukaryotic cell. But if we look at all the kinds of life out there, prokaryotic cells are actually 2 of the 3 domains, bacteria and archaea, while the remainder of the types of life that we are familiar with are in eukarya. Now, all of these things are still cells. So prokaryotic cells are going to have the same basic features as all cells. They have a plasma membrane, they have the material inside the plasma membrane, the cytosol, they have a chromosome that is made of DNA, and they have ribosomes that are going to make protein. Right? So again, here you can see the artist's rendition on the left of our typical prokaryotic cell, and then you can see TEM of that same bacteria. So you can see, again, now you start to see how we build pictures, artist renditions from the TMs, and it's not always obvious. So the challenge has been there when we look at these beautiful drawings. Right? Now, what is the biggest thing to remember about prokaryotic cells? The fact that they are simple and small, that they do not have a nucleus, they do not have membrane-bound organelles. The DNA is kind of coalesced in a nucleoid region, but we think of prokaryotic DNA as being naked as compared to the protein-bound DNA of eukaryotes in the nucleus. So the cytoplasm is bound by the plasma membrane, but that's pretty much it. So we're going to think of the prokaryotic cell as a studio apartment. You open the door to the studio apartment, and everything that you do and live in is right there. The kitchen, the living room, the bedroom, it's all right there. There are no rooms. There's no partitioning of the cell. Now, this is something you should become familiar with. This is a diagram you should be able to sketch out, and all the different components of this prokaryotic cell are listed on this next slide. So the fimbriae, or the pili, short hair-like structures for adhesion. Some bacteria have these, some do not. All have ribosomes. This is the site of protein synthesis. All have a plasma membrane. Most have a cell wall. And this cell wall in most bacteria is made of a substance called peptidoglycan. Now, based on what you know and the language that we learned when we were talking about biomacromolecules, can you suppose what peptidoglycan is made out of? What two biomacromolecules? I'll let you think about that. Then some bacteria have a capsule. Some bacteria, the capsule is quite extensive. It can be very, very thick, this mucus-like layer. This contributes significantly to what we call biofilms. So biofilms are when bacteria get together, they kind of coalesce under this mucilaginous layer, and they become very, very resistant to antibiotics and to efforts to remove them. Now, the flagella then is also optional. Some bacteria have these, some do not, but it's a long whip-like structure for locomotion. So you should be able to take those terms certainly and apply them to a sketch of a prokaryotic cell. Again, prokaryotic cells, simple, studio apartment, that's all we need to know.